Welcome to A1 Healthcare YouTube channel. Pfizer vaccine final results. It's highly protective, but how long for? The COVID-19 vaccine developed by Pfizer and BioNTech has completed clinical trials and is now being distributed worldwide. The vaccine has been issued temporary or emergency use authorization by regulatory bodies in the United Kingdom, Canada, and the United States. This is a watershed moment in history. It is now possible to build a biological shield against the virus. A highly efficient vaccine, when combined with existing physical obstacles, gives reason to believe that the pandemic can be ended. And after the tantalizing interim results released by Pfizer last month, we can now see the full peer-reviewed results of its Phase 3 trial. Here's what they tell us. Safety and efficacy confirmed. Approximately 37,000 people were included in the trial's safety analysis. Half received 2 doses of the vaccine, the other half a saline placebo injection. The vaccine was also tested on people who were at a higher risk of contracting COVID-19. About half of the participants were over 55, with 1/3 being overweight and another third being obese. People with pre-existing conditions that make them more vulnerable, such as diabetes, pulmonary disease, or HIV, were also included. However, the vaccine was tested in some groups more than others. The majority, 83%, of participants were white, and most of the trial, 77%, occurred in the USA, with additional participants in Argentina, Brazil and South Africa. As is common, pregnant women were excluded, and will likely be excluded from vaccination programs too, until we understand whether these vaccines are safe to use during pregnancy. Nevertheless, the safety profile of the vaccine is good, across different ages, ethnicities, both sexes and in individuals with pre-existing diseases. After being immunized, some participants experienced side effects such as headaches, nausea, or discomfort at the injection site. The majority of these reactions were mild to moderate, and they went away in 3 days. For at least 2 months after the second immunization, no more reactions were recorded. Analysis of over 36,000 individuals was used to calculate the vaccine's efficacy. 9 vaccinated participants became infected with the virus, compared with 169 individuals injected with the placebo. This equates to 95% efficacy. Most importantly, protection was high across different groups, regardless of age, ethnicity or underlying health conditions. Some participants became infected in between taking the first and second doses, highlighting the need to get the second dose. Efficacy after just the first dose was only 52%. If you take both doses, it's very likely you'll be protected from COVID-19, at the very least in the short term. Overall, the vaccine's effectiveness was verified, and its safety was thoroughly reported in this study. This isn't to say that the thesis predicts what will happen in the real world. We can't assume that the experiences of 19,000 people who have been vaccinated would apply to millions of people. It's impossible to detect less common side effects, for example. This is why very close monitoring of the vaccine now needs to happen as it rolls out, and authorities will need to rapidly respond if people have unexpected reactions to it. Decisive action has already been seen in the UK in response to previously unseen side effects in people with a significant history of allergic reactions. Similarly, it's possible that the efficacy of the vaccine in the real world, what we call its effectiveness, may also decrease, as it is used in more diverse populations and over longer time periods. And there are still key questions that need to be answered, particularly around the length of protection the vaccine will offer. It's almost certain that the immune response initially generated will wane over time. We don't yet know the lowest amount of immunity that needs to be retained to protect against infection, nor what type of immunity provides this protection. If vaccine-induced immune responses, such as antibodies or T cells, can wane to very low levels but still prevent infection, then this vaccine will protect people for a long time. But if immune responses must be constantly kept high for protection, it won't. We currently only have two methods for determining which is the case. The first is to continue to track the vaccine's effects in clinical trial participants. However, in order to get a reliable response, people in the study's unvaccinated, placebo arm would have to continue to participate, which raises ethical concerns. How do you strike a balance between the need to keep a placebo cohort and the rights of all patients to receive a good vaccine? According to the trial plan, following vaccination, follow-up could last for 24 months. This balance might be achieved by initially prioritizing vaccination for the most vulnerable placebo participants, and aiming to persuade the less vulnerable participants to remain in the trial. But if huge numbers of participants leave the trial, then the robustness of the analysis will deteriorate. We would then never know with good confidence how well this vaccine works over time. The second method will be to expose people to SARS-CoV-2 in a controlled environment and see what happens, known as human infection studies. Such trials are being planned in the United Kingdom, and they should be very useful in determining the levels and types of immunity required to protect against infection over time. If you liked this video, hit the subscribe button to receive more like it in the future.